Hi everybody, welcome to AP Biology, and today we are going to review the Hardy-Weinberg principle together. And you have some material that I had given out for homework on the Hardy-Weinberg principle. And in the lab manual on page 113 as well. I will be going over that in a separate presentation. This is sort of a review of the notes. And so a couple of things to keep in mind as we talk about the Hardy-Weinberg principle is sometimes it's known as Hardy-Weinberg Equilibrium. And what a Hardy-Weinberg equilibrium refers to is the fact that in an ideal situation, a population that is not evolving, a population that is not influenced by outside factors, so in other words, a fictitious population, that the number of individuals who are homozygous dominant, when added to the number of individuals that are heterozygous, and added to the number of individuals that are homozygous recessive, should always equal 100% of the population, or 1. And the reason why we use 1 is because all of these frequencies that we see here are in decimal form, and we have to keep that in mind when we're doing calculations. And I'll show you an example of a calculation in a little bit. So the Hardy-Weinberg equilibrium was named after two statisticians who were interested in trying to simulate what happens to the frequencies of genes, alleles, and genotypes in a population if we remove all of the factors that influence evolution. There are a few factors that we need to talk about that affect alleles and genotypes. Now, again, the difference between an allele would be something like this, whereas a genotype is something like this. And so what Hardy and Weinberg said is that if the frequencies of alleles and genotypes in a population are in equilibrium, in other words, they're not evolving from generation to generation, that the only way this can occur is if we neutralize specific disturbing influences. So some of the things that we would consider in an ideal circumstance where no one is evolving, no one is, or no population, because you can't evolve, a population evolves. No population is evolving. We have to eliminate a few things. We have to eliminate non-random mating. Non-random mating means you choose your mate because they're sexy. It's the sexy son hypothesis. It's runaway sexual selection. In other words, it's like the birds who are really attractive to other birds because they have beautiful plumage. That's a disturbing influence. And in the Hardy-Weinberg equilibrium, we have to eliminate that. In other words, anybody you bump into, you mate with. That's random mating. And so Hardy-Weinberg wants random mating. It doesn't want non-random mating. Hardy-Weinberg also tries to eliminate the factors involved in mutations. Now this is a tough one because mutations are generally caused by environmental factors or caused by DNA polymerase, which makes one error every 1 billion nucleotides that it complementary base pairs. We have mutation built into our system. So Hardy-Weinberg says, no, pretend like that doesn't happen. That's really hard to prevent because you can't prevent genetics from doing mutations. It just happens. The other thing the Hardy-Weinberg equilibrium tries to neutralize is the idea of natural selection. There should be no natural selection. So that means those three kinds of natural selection, directional and disruptive and stabilizing, can't happen. So there's no differential reproductive success. Nobody's more successful than somebody else at surviving, passing on their traits, getting food, avoiding predators. Everyone's equal, and that we know is not true. Hardy-Weinberg also tries to neutralize the effect of random genetic drift. Random genetic drift is a change in genes due to chance, and a change in genes or gene frequencies due to chance usually occurs by some kind of natural disaster or some sort of density independent limiting factor, a volcano or an earthquake or an asteroid or something that just wipes everybody out. That's random genetic drift. Well, Hardy-Weinberg says, no, you can't have that. Hardy-Weinberg also says there's no gene flow. Well, gene flow is migration. So in other words, everybody stays exactly where they are, and the population is infinitely large and doesn't change. So there's a factor that says you can't move. Big population, stay exactly where you are. That doesn't happen in real life. The other disturbing influence that we cannot have, according to Hardy and Weinberg, is meiotic drive, variation of your gametes, which is due to a few things. We can't have crossing over, and we can't have independent assortment. Now, again, all of these things that I've just mentioned, well, they happen. They happen in nature. Hardy-Weinberg says, no, we don't want this to happen because we just want to track the genes. So, these disturbing influences that we just mentioned are always in effect in a real population. But what Hardy and Weinberg said is that in an ideal state, we neutralize all of those disturbing influences so we can analyze the changes in genes in a population without the effects of evolution. This is great for tracking mutations in human populations. This is great for determining how many carriers there are for certain genetic disorders. That's important. But really, we can't apply this beyond that in real life. So, again, the assumptions of Hardy-Weinberg: no natural selection, no sexual selection, no directional selection, no disruptive selection, no stabilizing selection. In other words, it really doesn't matter what you look like. All three of these little creatures that we see here all have an equal opportunity at surviving, according to Hardy-Weinberg. In addition, he says no mutations, no changes in your DNA. Well, that's kind of tough because mathematically that's going to happen regardless. He says there's no migration, or they say, Hardy and Weinberg. So in other words, these predators can't make their way over here to knock off their prey. That's no selection and no migration. They're not allowed to move. And if you're not allowed to move, well, that's one of the assumptions that Hardy and Weinberg allow for their equation. The other thing that they mention is you have to have an infinitely large population. Well, that means there's no isolation. And if there's no isolation, then that doesn't allow organisms to evolve into separate species. Remember, we said geographic isolation leads to all those reproductive isolations, temporal isolation, for example, or behavioral isolation, or hybrid isolation, and that's what really leads to speciation. Hardy and Weinberg don't want that. They want to eliminate those factors. Finally, they allow for random mating. Random mating means everybody has an equal opportunity to mate with each other. So if you bump into somebody else, you can mate with them. Doesn't matter what you look like. Everyone is fair game. And we know in nature that doesn't happen. Organisms look for the traits in their mates that they want to see in their offspring. That's the sexy son hypothesis. And so what we need to keep in mind when we're looking at all these letters and values is that these all are going to be in decimal form. And the reason why we do this in decimal form is because we're talking about frequencies. We're assuming a few things with Hardy-Weinberg. We're assuming that we have a large population, there's no selection, no migration, no genetic drift, no natural disasters, and you can't choose your mate, it's just you bump into something and you mate with them. And so one of the other assumptions that Hardy and Weinberg make is that everybody starts off heterozygous in a population. In other words, if I were to come over here to my Punnett square and I do the cross between two heterozygotes, that I get a 1 to 2 to 1 ratio of dominant to heterozygote to recessive. And so 25% to 50% to 25% ratio gives me a total of 100% of my population. I have 4 boxes. I have 4 total kinds of outcomes. And so that ratio should equal a 1 whole population, 100% of the population. So all Hardy and Weinberg did was they assigned these alleles a letter. And they said, all right, little p is the dominant allele. Little q is the recessive allele. That's an allele. Remember, an allele is a gene. Then they said, okay, well, p squared must be t times t, which is p times p. Well, that's a genotype. That's kind of what you are genetically. It takes 2 alleles to make a genotype at least. And so if we're talking about a genotype, we're talking about what you end up looking like. And so genotype is very similar to what we call phenotype, what you look like. So your genes determine what you look like. The alleles are the actual genes that have been separated. This is little t, little t. 2pq is the heterozygous condition, and this 2 over here comes from this 2 over here because I have 2 heterozygotes. And so I have 2 big T, little t's, because that represents 50% of my population. If I take all of the dominant alleles and I add them to all of the recessive alleles, I get 100% of all the alleles in the population, or 1, since we're using decimals. So 0.5 plus 0.5 equals 1 in this example. Finally, if I put all of this together and I say, okay, my dominants plus my heterozygotes plus my recessives equals 100% of all my genotypes in the population, well, this comes from this, which is this, which is this. They're all related. And so these equations will always be given to you on a reference table. There's nothing to memorize. You just have to learn how to plug and play. That's the important part of this. So again, to review, this is the allele equation. And we usually use this first. This is the genotype equation. And we usually use this second to solve for problems. Together, and especially this one, this is called the Hardy-Weinberg Equilibrium. And a population is in equilibrium when there's a non-evolving population. In other words, we have to keep all of those conditions that Hardy and Weinberg mention constant. Eliminate selection, eliminate sexual selection, eliminate mutations. This isn't possible. We know that. This is just a mathematical exercise. It's like the ideal gas law. It's kind of fake, but we do it to track genes. I kind of like this graph because it actually shows us a little insight into this while using a graph to describe and also to help us calculate Hardy-Weinberg equilibrium. Let me show you what I mean. We have our three conditions. We have in orange, we have our recessive, homozygous recessive, line. Here in green, we have our heterozygotes. And here in blue, we have our homozygous dominants. If I take my little a population right here at 0.8, okay, this means 80% of my population is little a. And I follow this line down, and excuse my line for not being perfectly straight, but I go right to here, and I see, oh, my heterozygotes make up about 18% of the population, right around there. And I follow this down, and I go, ooh, down to the blue line, and I say that my dominant individuals make up 2% of the population. If I add up 2%, which is 0.02, plus 18%, which is 0.18, plus 80%, which is 0.80, this is p squared, this is 2pq, and this is q squared. Guess what they equal? That's a non-evolving population. Try it with this graph. Pause the video if you want and do it along some other set of lines. Take those numbers and estimate what those values are, or estimate what these values are, maybe right here in the middle and right here where they cross, and add up these points. Add up these points and see what you get. It should be 1 or close to it because we're estimating. I'll give you a chance to do that, or you can pause right now and you can do it yourself. Finally, this is a question we did in class. And so in order to do a question like this, we always have to start with a few steps. The way to solve a question that's a Hardy-Weinberg question is we always try to find our q squared, which is almost always given to us. Then we find q, which is taking the square root of q squared. Then we find p, and we find p by using this equation. And then we do what the question is asking us. Then we either solve for p squared or we solve for 2pq, depending on the problem. Well, in this question, they're giving me q squared. It's right here. They're telling me that q squared is the recessive genotype. It's 16%. They want me to figure out what is just the dominant allele and then what is the genotype, big A, little a. So they're giving me q squared. They want me to solve for p, and they want me to solve for 2pq. So the way that we do this is we take our q squared value over here, and we just write q squared equals 0.16. That's 16%. I'm going to take the square root of q squared to give me q. So that means I'm going to take the square root of 16% to give me 0.4. That means that 40% of the population has a recessive allele. I'm going to use this equation, p plus q equals 1, and I'm going to solve for p by subtracting Q from each side. And so P equals 1 minus 0.4. That means it's 0.6. This solves my answer over here. I just solved that part. Now that I know what Q and P are, I can put those values into this. 2PQ. 2 times 0.6 times 0.4 should be 0.48, or 48% of the population is heterozygous. If you're still unsure of how to do these questions, work on your practice homework questions on the handout that I gave you. Page 113 in the lab manual has about 5 questions you can continue to work on. The answers and the way to solve those questions are in separate videos that you can watch and try to solve for yourself and work on as we go along. You can always email me, you can contact me through Teams, or you can hit me up through Remind, and we can talk about it. I wish you good luck, and I guess I'll talk to you soon. We'll continue doing some of these problems. There's also your review book. See you next time.